Hi students, welcome to SPSVERSITY. Today we are going to discuss about biotechnology principles and processes. What is meant by biotechnology? So biotechnology deals with the techniques of using live organisms or enzymes from organisms to produce products which are useful to humans. So whatever the products we get from the live organisms which are useful to humans, it is called biotechnology. And we follow the definition of European Federation of Biotechnology. So what this EFB says? So biotechnology is an interdisciplinary branch of biology. So it is an integration of science, organisms, cells, and molecular analogues for products and services. So this is the definition of European Federation of Biotechnology, which we follow. And when it comes to principles of biotechnology, so there are two basic principles of biotechnology. One is genetic engineering. Another one is the maintenance of sterile environment. So what is meant by genetic engineering? So genetic engineering is the technique to alter means to change the chemistry of genetic material. And it includes to introduce these into host, that is, introduction of genes into organisms. Whenever a gene is introduced into organism, it will change the phenotype of the host organism. That is, the manipulation of genetic material, the introduction of genetic material into organisms, which will change the phenotype of host organism. So what is meant by sterile environment, maintenance of sterile environment? So sterile environment means it is the sterile ambience in process to enable growth of only the desired microbe. That is, whatever the microbe we are desired to grow, only it will enable the growth of microbe. Or eukaryotic cell in large quantities for manufacture of biotechnological products like antibiotics, vaccines, enzymes, like many other. So here only it will allow the growth of desired microorganism or a specific eukaryotic cell. Other than this, it will not allow any one. So it is called the maintenance of sterile environment. So when you come to the basic steps of genetically modifying organism, so there are three basic steps for modifying an organism. The first one is identification of DNA of desirable genes. Introduction and identifying DNA into host. Maintenance of introduced DNA in the host and transfer of the DNA to its progeny. So here you can see the picture after identifying the ligating there, that is separating isolation. The second step is introduction into the host. And third step, that is maintenance of introduced DNA in the host and transfer of the DNA to its progeny. So for these three steps, that is to carry out the process of the biotechnology, we require lot of tools like restriction endonucleases, gel electrophoresis, cloning vectors, competent host. Like this, these are the tools which are required for to carry out the process in the biotechnology. So the first tool is the restriction endonucleases. So what are these restriction endonucleases? These are enzymes which are responsible for restricting the growth of bacteriophage. Bacteriophage means the virus which will infect the bacterium, and these are isolated from Escherichia coli. What this enzyme will do? It will add methyl groups, that is deoxyadenosine methylase. So what this enzyme will do is it will cut the restriction it will cut the DNA. So these restriction endonucleases will cut the DNA. So what is the main purpose of this restriction endonucleases is to cut the DNA. And the first restriction endonuclease was HIND2. The name was HIND2. And later on, many restriction endonucleases were extracted like ECO-R1. So on what basis the name is decided? So what is the criteria for naming this or the restriction endonuclease? So here in ECO-R1, ECO means it is name of the species, that is Escherichia coli. R means the strain. Roman letter 1, it is the Roman number 1 indicating the order of enzyme which was isolated from the bacterium. If it is 2, it is named as ECO-R2. If it is 3, ECO-R3. Like that, in the sequence of enzymes, it is isolated. So this restriction endonucleases will recognize the palindromic sequence. What is this palindromic sequence? That is, in a DNA, double stranded DNA, if we read the two strands in opposite direction, that is, it will be same, like Malayalam. Suppose if we take Malayalam. So Malayalam, if you read in one direction, that is in this direction, it will be of same. At the same time, if you read like this, it will be of same, Malayalam. So this is called palindromic sequence. And this restriction endonucleases, what actually they will do? For example, if we take, so this is the palindromic sequence, GAATTC. So if you read in this direction, it is GAATTC. If you read in this direction, it is GATTC. So this, it will cut between G and A. The ECO-R1 will cut between G and A. So we will get a part of DNA with hanging single stranded one. So these are called sticky ends. These are called sticky ends. But whereas when you come to blunt end means both are cut at the same end, which leads to which there is no single stranded. That is, it is called blunt end. So restriction endonucleases will form sticky ends. So here you can see the ECO-R1. The ECO-R1 palindromic sequence is GAATTC. So if you write in opposite sequence GAATTC, so it will be, that is opposite sequence. So here you can see the ECO-R1. This is the enzyme ECO-R1, which will form on cutting, which will form sticky ends. Okay. So these sticky ends which are formed, as they have complementary DNA sequence, they can bind again. That is, in this gap, the foreign DNA is introduced, which leads to formation of recombinant DNA. Okay, so now by using restriction endonuclease enzymes, the DNA is made into fragments. Okay, so ultimately by the end of the action of restriction endonuclease enzymes, we got DNA fragments. And these DNA fragments are separated and isolated by a process called, based on size, that is called Agarose Gel Electrophoresis. So what is this Agarose Gel Electrophoresis? So it is based on principle, that is, the negatively charged nucleic acid molecules in agarose will move in an electric field, that is electrophoresis. The shorter will move faster, and whereas than the longer ones. The longer ones cannot move faster, whereas shorter ones can move faster. Okay. So here in gel electrophoresis, at a specific pH value, that is, it has anode and cathode. So under the influence of electric field, so these charged particles means the DNA being negatively charged, it will migrate towards positively charged, that is towards cathode. So depending upon the net charge. So DNA being negatively charged, it will migrate towards the cathode. So here electrophoresis is the movement of molecules by an electric current. Okay. So here nucleic acid moves from negative to positive pole. That means positive means the cathode, and DNA being negatively charged, it moves towards the cathode. Okay. So here you can see. That is, DNA when placed in electric field. So DNA is negatively charged. When placed in electric field, it will migrate towards the positive charge. So here in gel electrophoresis, it is connected to electric field. It is placed in electric field where the DNA will move towards the poles. Okay. So this is the gel electrophoresis. So you'll see what are the components of gel electrophoresis system. So one component is the power supply. So where the charged particles with a cathode and anode. Next, a buffer with a mixture of water and ions. Agarose gel, which is extracted from red algae, that is, in which the DNA will migrate. Gel casting materials. DNA ladder, which is mixture of different DNA fragments of known lengths. Loading dye, that is ethidium bromide, which contains dense material for visualizing the DNA stain, that is, which allows visualization of DNA fragments after electrophoresis. So here you can see different components. The power supply, okay, the electric leads, the cover, okay, and the gel tank where agarose gel is kept, the casting tray, and gel combs. So these are the equipment in gel electrophoresis, the technology which is used. Okay. After allowing, that is, after keeping in gel electrophoresis, the DNA, that is what you are seeing, the bands, the DNA is separated according to the size, like thick bands. Okay. So this allows DNA visualization. That is, after gel electrophoresis, it looks like this. Okay. After this, it is treated with, that is, dye, that is ethidium bromide. Okay. After treating with ethidium bromide, okay, that is, it is exposed to ultraviolet rays to view the DNA strands. Okay. When it is exposed to ultraviolet rays, the ethidium bromide will fluoresce, that is reddish orange, in the presence of DNA. So you can take a photograph with a digital camera. So for visualization of DNA, that is, we use the dye ethidium bromide, as well as we use ultraviolet rays, which is used for visualizing the DNA. So here you can see finally the picture, that is, all the large DNAs are present at one point, the small DNAs at one point. Okay. So these are molecular weight markers. Depending upon the size, their molecular weight markers are kept. So this is the photograph, that is electrophoresis. After completion of electrophoresis and applying UV rays, that is exposing to UV rays, you can see like this different bands of DNA. Okay. So what is the application of this gel electrophoresis? So the application is, we can estimate the size of DNA molecule, okay, which is used for restriction mapping of clone DNA. Analysis of PCR products, that is for genetic diagnosis and genetic fingerprinting. So separation of DNA fragment, that is extraction and purification. So separation of restricted genomic DNA or RNA prior to the next step, that is, in DNA the next step is southern transfer and next step in RNA is northern transfer. Okay. So follow us on SPSVERSITY and social media. Till now we discussed about the restriction endonucleases, and next we discussed about gel electrophoresis. And next we are going to discuss about cloning vectors, which is an important tool in the recombinant DNA technology. So what is the use of vectors? So these vectors are used for preparing recombinant DNA. So what is recombinant DNA? That is, inserting DNA of our choice into the host DNA. These which are known as cloning vectors. Any cloning vector, it should have some specific features. That is, these features are, it should have an ORI, origin of replication, selectable marker, cloning sites, and vectors for cloning in plants and animals. First we'll see what is this origin of replication. So in this origin of replication, that is, when you come to origin of replication, replication means this is the sequence from where the replication starts. From where the replication starts. It is called, in short, it is called ORI. Okay. So any piece of DNA with linked to this sequence, it can be replicated within the host cell. So this sequence is also meant for controlling the number of copies of DNA. Okay. So here, whatever the number of copies, so if many copies are required, vector must have high copy number. So ORI, this is origin of replication, and these are the enzymes which are associated. This is primase, helicase, and these are single standard binding proteins which will bind to the separated DNA strands, which are These are the enzymes and proteins which are useful during the process of replication. So here you can see this is the point where the replication starts. It is called origin of replication, and this is called replication fork. On either sides we can see the replication forks. Okay, so this is ORI. And next, when you come to selectable marker. So in this selectable marker, what is the purpose of selectable marker? The selectable marker is used for selecting successful transformation. Okay. So in a vector, what are selectable markers? So generally in a vector there will be generally antibiotic resistant genes are used as a selectable marker. Okay. So here this is a artificially prepared, that is PBR 322. So which is an cloning vector which is generally used in genetic engineering, recombinant DNA technology. So here you can see, so this is ampicillin resistant gene, and this is, that is tetracycline resistant gene. Okay. This is tetracycline resistant gene. And this is the origin of replication. This is the place sequence of origin of replication, which in short it is called ORI. So this plasmid has gene and antibiotic resistant genes, added genes. So generally antibiotic resistant genes are used as, that is the what we call, that is selectable markers. Okay. So here in selectable marker, there is inside the selectable markers, again there are specific parts, they are called cloning sites. That is, what is cloning site? That is where DNA to be cloned is inserted. It is called cloning site. Okay. So here you can see there is usually a restriction enzyme present in any of the selectable marker site. Okay. For example, if we take, this is a cloning vector PBR 322. So these are selectable markers, tetracycline resistant and ampicillin resistant. In each, that is selectable marker, there is an restriction enzyme. Like for example, if we take tetracycline resistant BAMH1 SAL1, in ampicillin resistant gene, that is PST1 PVU1. So these are the sites, these are called cloning sites. So this cloning sites which will cut, this is the point where restriction enzymes will cut the DNA. Okay. So here ORI means origin of replication, or OP means replication of proteins. The proteins which are required for replication, these arise from this point. So here at this point, that is where the DNA is cut with the help of this specific restriction endonuclease. So that we get, that is, so that we have this, that what we call where we get this, that foreign DNA is inserted. So this is the point where the foreign DNA can be inserted. Okay. So here when foreign DNA is inserted, whatever it is, okay. So this, whenever foreign DNA is inserted in a specific gene, whether it is an ampicillin resistant gene or tetracycline resistant gene, it becomes inactive. It is called insertional inactivation. Okay. So inactivation is due to the new insert. Okay. So this property is used to find out the recombinant and non-recombinant cells. So here you can see in the picture E. coli. In this E. coli, the recombinant plasmid is introduced. It is called transformed cell. Okay. Whereas when you take this non-transformed cell where there is no recombinant plasmid, where a transformed cell there is a recombinant plasmid. Okay. So it is grown in a culture plate. So here you can see, that is, when it is grown overnight, we can see some white colonies, whereas we can see some blue colonies with only PUC19. That is, these are ampicillin resistant colonies. That means they are able to tolerate ampicillin resistant. That means in ampicillin resistant gene, that is, foreign DNA is not inserted. Okay. So here when you come to this one, when a foreign DNA is inserted into, that is, a gene, the gene becomes inactive. That is called insertional inactivation. So this inactivation of the genes, it is used to identify the transferments and non-transferments. Okay. When you come to cloning vectors in plants and animals, in plants there is specific, that is in animals there is specific. In plants generally a bacteria is used, that is Agrobacterium tumefaciens. This is a bacteria which is generally used in plants. So this Agrobacterium tumefaciens, the plasmid which is present, it is called TI. TI means tumor inducing. So this tumor inducing plasmid, due to this it will cause crown gall. Okay. This, the Agrobacterium tumefaciens of the wild type which has TI plasmid, which will cause crown gall in dicots. So through this Agrobacterium tumefaciens in plants, the foreign DNA, that is recombinant DNA, is inserted into this. Okay. So here you can see this is this Agrobacterium tumefaciens. So this is tumor inducing plasmid. Okay, where foreign DNA is inserted. So which is inserted into this plant. So which will form, this is crown gall. Okay, and here you can see the transformed cell. Okay, this is the method in the what we call, that is, in plants. Okay, and at the same time there are some other techniques. Specifically when we come to, that is non-viral gene delivery, that is, here there is, that is, the plasmid DNA, that is, it is introduced into the host cell by hydrodynamic injection or by electroporation. Okay. In animals it is by retroviruses or adenoviral vectors. Okay. So there are different methods of introduction of rDNA into host. In plants we use Agrobacterium, whereas in animals we use retroviruses, which generally cause cancers in animals. Besides retroviruses and the Agrobacterium tumefaciens, there are other methods like heat choke, micro-injection, ballistic method, and use of disarmed pathogens. What are these methods? In heat choke, the cell is subjected to high temperatures, at the same time it is subjected to low temperatures. Okay. This is how, during this process, what happens is the permeability of the plasma membrane will increase. And next, micro injection, which is generally applied to animal cells. Okay. Where by taking a fine needle, it is injected into the animal cell. Ballistic method, which is called also gene gun, which is generally used in plant cells. where here the tungsten or gold particles which are used as a which are used to introduce into this. So on the surface of this gold or tungsten, the recombinant DNA is attached to this and it is shooted into plant cell. And use of disorbed pathogen, that means pathogens, even though they are pathogens, by removing the genes which cause diseases, it is called disarmed pathogens, by which the recombinant DNA can be inserted into host. So these are different methods of introduction of recombinant DNA in host: heat shock, microinjection, ballistic method, use of disorbed pathogens. Okay. So welcome to SPSVERSITY follows on social media. So till now we discussed about different tools which we use in recombinant DNA technology. That is restriction endonucleases, gel electrophoresis, and next is cloning vectors, where we use different methods to introduce into. Next one is methods to introduce recombinant DNA into host. Okay. And when you come to this process, what is the process of recombinant DNA technology? By using these various tools. In this process, the first step is isolation of genetic material, that is isolation of DNA. Next, after isolation, and that is the separation, that is after isolation, the cutting of DNA at specific location by using restriction endonucleases. Amplification of gene of interest. Amplification means multiplication, making many copies of the DNA. And insertion of recombinant DNA into the host, and later on obtaining foreign gene product from this recombinant DNA. So for this isolation of genetic material, there are different ways, that is where we use different enzymes for different cells. So to extract the DNA from bacterial cell, we use an enzyme lysozyme. And to extract DNA from plant cells, we use enzyme cellulase. From fungal cells, we use chitinase. Okay. And to digest the plasma membrane, we use proteases and lipases. Okay. Ultimately, that is the ribonucleases, which will digest the what we call the RNA, which is associated with DNA. And the proteins which are associated with DNA, they are digested by proteases. And later on, to this, it is added, that is the ethyl alcohol is added to this. Chill ethanol is added. So after chilled ethanol, adding of chilled ethanol, we can get the DNA by spooling. The process is called spooling. Okay. So like this, we use different enzymes for different cells. That is, to break bacterial cells, we require lysozyme. Plant cells are made up of, that is, the cell wall of plants is made up of cellulose. To break this, cellulose is required. To break fungal cells, we require chitin. Fungal cells, if cell wall is made up of chitin, the enzyme which we use is the chitinase. And to digest the plasma membrane, we use proteases and lipases. Plasma membrane is made up of proteins and lipids. And whatever the proteins and the RNA associated with this DNA, like to digest the proteases and ribonucleases. And finally, to this chilled ethanol is extracted, where we get a pure DNA. By spooling, we get the DNA. Okay. So by using various enzymes, ultimately we have either is separated, isolated DNA. Okay. So this isolated DNA, whatever we got after isolation, the next step is cutting off DNA at specific point by using restriction endonucleases. So here, after cutting, amplification of the desired gene by PCR, preparation of recombinant DNA, that is insertion of recombinant DNA into host, obtaining the foreign gene product. Okay. So here in this, if you take this, that is restriction endonucleases, which will cut the DNA at specific which will cut the DNA at specific sequences, that is what we call, that is palindromic sequence. Like as we discussed in restriction endonucleases like ECOR1, HIND2, these all are different enzymes. After this, the amplification of desired gene by PCR. Amplification by PCR means PCR full form is polymerase chain reaction. And in this polymerase chain reaction, so there are basically the different steps, 3 steps in this, where the DNA is made into millions of copies within 1 hour. That is denaturation. Okay. Next step is annealing, and the final step is extension. In denaturation, the DNA is kept at 90 degrees so that both the strands of DNA get separated, and it is brought to 65 degrees centigrade at annealing so that DNA primers are formed. In extension process, that is the temperature is at 72 degrees centigrade. That is, if you take the entire process of polymerase chain reaction, it is carried out at high temperature. So in this high temperature, DNA polymerase cannot function. So that's why the DNA polymerase is extracted from a bacteria, that is Thermus aquaticus, which is used to, that is the Taq polymerase, which is used in this process. So that the specialty of this enzyme is even it can function at high temperature. Normal DNA polymerase cannot function at high temperature. Taq polymerase can function even at high temperature. It is extracted from a bacteria which lives in high temperature, that is Thermus aquaticus. That's the name Taq polymerase. Taq means Thermus aquaticus. Okay. The desired gene is made into many copies. Okay. And this many copies, whatever the desired gene is made into many copies, this is introduced into, that is vector. By using vectors, the foreign DNA is inserted into vector. Then it will become a recombinant DNA. So this recombinant DNA or DNA is introduced into the host by using various methods. Whether it is by gene gun, bioelastic, micro-injection, desorbed pathogens, like this various methods are used. Okay. So ultimately the recombinant DNA is introduced in the host. So where we get ultimately the recombinant DNA is introduced to the host. Okay. After introducing into the host, how to obtain foreign DNA product? For obtaining foreign gene product, we require bioreactors. What is the purpose of bioreactors? That is to produce in large quantities. So appreciable quantities of product to produce in large quantities. Okay. So this is just an picture image of bioreactor, from which we can produce various products like vaccines, antibiotics, hormones, enzymes, proteins, which are used in various purposes. These all are extracted. Okay. So here you can see the process. Okay. So in this process, you can see that is the cloning vector, the plasmid, which is used as generally in most of the biotechnological process. The plasmid is used as a vector. So by using restriction, same restriction endonuclease enzyme, the foreign DNA is cut along with the same restriction endonuclease enzyme is used to cut the plasmid also. So wherever the foreign DNA, that is the plasmid because where the foreign DNA is inserted, that is rDNA. It is called recombinant DNA. And this recombinant DNA is inserted into host. That is, it is called transformed cell. Transformed cell means it is host cell with rDNA. What is meant by transformed cell? That is host cell with recombinant DNA. So here you can see these are host cells with recombinant DNA. So these are colony of transformed cells. So this, whatever the foreign DNA is inserted, it will show the expression of the protein. Whatever it is meant for that, it will show the expression of protein. So here you can see the images of bioreactors. So this is first one, that is simple stirred tank bioreactor. This is second one is sparged stirred tank bioreactor. So if you see the parts here, that is simple stirred tank bioreactor. There is a motor. So there is a culture broth where nutrient are added. So this is flat blade impeller, foam breaker. This is pH controller. This is steam for sterilization, that is to prevent the growth of microorganisms. And this is the port through which sterile air is sent. Okay. So various ports are there through which the sterile air, the pH control, the steam for this, these all are maintained. Still more advanced beyond the simple stirred tank bioreactor is sparged stick tank bioreactor. So where we can see here, there is a, this one is rotor. Okay. So here you can see this is increased area for oxygen transfer. Okay. So which will bubbles will dramatically increase the oxygen transfer area. So sparged stick stirred tank bioreactor is more advanced than simple stirred tank bioreactor. Okay. So whatever we got, that is the product, that is either by in the bioreactors, whatever we get. Okay. To get the products from bioreactors, we should add raw materials into the culture broth. So the culture broth to which the raw materials is added. And inoculum generation, that is the transformed cells, we should add to bioreactors. This is called upstreaming process. After upstreaming process, after bioreactor, after the processing in bioreactors, next you should separate biomass separation, that is product purification and product finishing. These three steps are called downstreaming process. That is after bioreactor, after the processing in bioreactor is completed, the next step, that is product purification, biomass separation, product finishing, these all are called downstreaming process. And the steps which are above the bioreactor, that is adding of raw materials, inoculum generation, the pH control, supplying of sterile air, these all the conditions which we provide to bioreactor, these all will come under upstreaming process. So already we discussed about the simple stirred tank bioreactor and sparge stirred tank bioreactor, where different types of bioreactors are used for different types of products. So from bioreactors we can extract various products like vaccines, the proteins, hormones, okay, like enzymes which are used in various industries. These all are extracted the products. So whatever the products we extract from these bioreactors, so they should be separated, purified and isolated. So this separation, purifying, that is finishing, this all will come under downstreaming process. So what actually the process, downstreaming process is? Like the separation, purification of biosynthetic products before it is released into market as a finished product. It is called downstreaming process. That is, as and when we get the product from bioreactors, it cannot be released into market because it is not a purified product. So lot of things will be attached to the product. Why? Because it is kept, it is obtained from bioreactor. So to overcome this, we do downstreaming process. In downstreaming process, there will be separation, purification of this, and finally it is released into market. Okay. What are the different steps here? That is, the formulation of product with preservatives, additives. That is, whatever the product we add, or whatever the product we are preparing, for storage purpose we should add, that is, preservatives, or sometimes for giving a good flavor we should add flavors, flavonoids like this. So there should be a formulation for this. So for formulation of the product like preservatives, flavonoids, these all, they should be added in proper proportion so that we get ultimately a finished product. So what preservatives, what kind of preservatives, what is the quantity of preservatives, what is the proportion, so that they can be stored for a specific period of time. Okay. After this formulation of product with preservatives, the next is clinical trials of drugs for our formulations. So whatever we got, we should go for example, if it is a medicine or vaccine or anything, it should be gone through clinical trials. So different phases of clinical trials, where it is done on mouse and various, in later stages it is done on humans. So when it is done subjected to clinical trials, whether it is a medicine, drugs or resource, after getting the required the result, the next thing is strict quality control on testing of each and every product. Okay. So strict quality control testing. So quality controlling is required for each and every product, so that there should be no compromise in the quality. So whatever we prepare the products, so these products should be added with preservatives, that is the formulation of preservatives, the formulation of different components to store. And next is clinical trials. Clinical trials means whether it is a medicine or whatever it is, drug or vaccine, it should be first tested on the primate, that is mouse like this. Later on it is human trials. So which is a lot of processes there, in different phases are there in clinical, like phase 1, phase 2, phase 3 trials. And the quality control, the testing of each and every product. Okay. So we should test the each and every product for quality. After all these it is called a finished product. Okay. And this finished product should be packed properly. So there should be a proper packing process. It cannot be rigid like anything. So there should be a packing. So not only the formulation of preservatives, clinical trials, quality control, even packing in a proper way, the way we pack depending upon the things. Okay, the labeling one, that is the labeling, the date of manufacturing, the expiry date, best before, these all should be added on the packing content. Okay. So these all products, these all things are done before it is sent into market. That is before it is released into market. So it has to undergo various processes in downstreaming. That is, in downstreaming process, that is not only separation, purification, it also involves, that is, formulation of product with preservatives. Next, clinical trials of drugs and formulations. That is, you should, if it is a medicine, it should be gone through proper clinical trials, and proper, that is, permissions should be taken from the prior authorities. So for releasing to market, these permissions from specific authorities should be taken. Okay. Later on, strict quality control testing. After this, we'll get a finished product. After this, the next one is packing, the labeling, and next it is released into market for consumers. So this is how we get the product after downstreaming process. So in this we discussed about the tools which we use in biotechnology, like restriction endonucleases, gel electrophoresis, next cloning vectors, and we discussed about the process of recombinant DNA technology, that is isolation of DNA, next the cutting of DNA, amplification of DNA by Pcr, where we discuss the 3 steps in Pcr: denaturation, annealing, extinction. Next, inserting of the foreign DNA into that is making into recombinant DNA, inserting of recombinant DNA into host, and next obtaining foreign gene product from bioreactors, where we discussed about simple stirred tank reactor and sparge stirred tank bioreactor. And upstreaming, after upstreaming process, that is after bioreactors, that is whatever we get product, that is bioreactors, that is next we discuss about downstreaming process. So this is how we get products from the, that is the biotechnology process. Thank you all. Follow us on social media. We'll meet in the next session.